Welcome to The Explainer. Today, we're diving into a high-stakes, microscopic relay race. It's a wildly complex process that is happening inside your body millions of times a second, literally right at this very moment. We're talking about neural conduction and synaptic transmission. This is the biological machinery behind absolutely every thought, action, and feeling you have.
So think about this for a second. Why does a simple text message from your roommate saying, "Wake up, you're late for your exam," instantly produce that awful sinking feeling in the pit of your stomach? I mean, how does visual information hitting your retinas translate into an immediate sensation of panic and then an overt physical scramble to get dressed and run to class? Well, the answer entirely involves the incredible activity of your neurons translating information into physical action. Let's get into it. To really understand this, we need a roadmap. We're going to break this incredibly fast journey down into 5 chronological milestones. First, the resting potential. Second, grading the signals. Third, hitting the firing threshold. Fourth, the action potential itself. And finally, crossing the synaptic cleft. Okay, step 1, the resting membrane potential. This is all about the primed neuron. You see, when a neuron isn't actively firing, it's not simply turned off. It actually maintains a state of intense baseline tension. It's primed and loaded, exactly like a pulled bowstring, just waiting to fire an arrow. And this delicately balanced tension has a very specific number attached to it, negative 70 millivolts. In its resting state, the neuron maintains an electrical gradient, which we call polarization. Basically, this just means the inside of the cell has a negative charge compared to the outside. So how exactly does it maintain this negative charge? Well, it's a tense standoff, and it's all about the distribution of ions across the cell membrane, which acts as a very selective gatekeeper. Think of it as the insiders versus the outsiders. The intracellular fluid, the inside of the cell, contains high concentrations of positively charged potassium ions, but they're hanging out alongside negatively charged proteins that drag the overall charge down. Meanwhile, the extracellular fluid on the outside is just swimming with positively charged sodium and negatively charged chloride ions. Now, because of concentration and electrical gradients, sodium absolutely desperately wants to rush into the cell. But at rest, the membrane's sodium channels are tightly closed shut. To keep this negative 70 millivolt tension perfectly set, the neuron uses an active transport mechanism called the sodium-potassium pump. This pump is constantly shoving 3 sodium ions out of the cell for every 2 potassium ions it pulls back in. It's working so hard, in fact, that this tiny little pump consumes a massive percentage of your brain's total daily energy just to keep that bowstring pulled back. Which brings us to step 2, grading the incoming signals, receiving the messages. Our primed neuron is constantly receiving a barrage of chemical whispers from its neighbors. These chemical whispers create brief, local electrical changes called graded potentials, and they spark a constant debate inside the cell. These incoming signals essentially fall into 2 main categories. First, we've got EPSPs, or excitatory postsynaptic potentials. These are your go signals. They cause a little sodium to enter, making the inside of the cell slightly more positive, pushing the cell closer to firing. On the other hand, we have IPSPs, inhibitory postsynaptic potentials. These are stop signals. They cause potassium to leave or chloride to enter, making the inside even more negative, actively pulling the cell further away from firing. Moving on to step 3, reaching the firing threshold. It's time to tally the votes. With all those go and stop signals flooding in simultaneously, the neuron has to act as the ultimate decision maker. It has to tally up the votes. We call this vote tallying process neural integration, and it primarily happens at a cone-shaped segment of the cell called the axon hillock. The neuron sums up the signals in 2 ways. Temporal summation occurs when several impulses arrive from a single neighboring neuron in really quick succession, building upon each other over time. Spatial summation, on the other hand, occurs when impulses arrive simultaneously from several different locations across the neuron surface. Now, if the summation of all those excitatory go signals manages to overcome the inhibitory stop signals and it depolarizes the membrane from negative 70 millivolts up to about negative 55 millivolts, well, the cell reaches its threshold of excitation. Negative 55 millivolts is the magic number here. Once this specific threshold is hit, it triggers an unstoppable, irreversible directive to fire. There's no going back. And that kicks off step 4. Firing the action potential, the point of absolutely no return. We have officially passed the point of no return. The neuron is now sprinting down the track, and it operates strictly on a principle called the all or none law. This law states that the amplitude and velocity of an action potential are totally independent of the intensity of the stimulus that triggered it. Once that negative 55 millivolt threshold is crossed, the neuron fires completely, or it doesn't fire at all.
To really lock this concept into your mind, think of flushing a toilet. You know you have to push the handle with at least a certain amount of strength to trigger it. That's your threshold. But if you push the handle way, way harder than necessary, the toilet doesn't flush any bigger or any faster, right? The action of the flush is exactly the same once it's triggered. Neurons work the exact same way. So what does this actually look like? Well, notice the dramatic voltage spike on the graph. When the threshold is reached, boom, voltage-gated sodium channels burst open. Remember all that sodium anxiously waiting outside? It suddenly rushes in, causing rapid depolarization. The voltage shoots all the way up to positive 30 millivolts. At the very peak, the sodium gate snaps shut. Then, potassium channels open up, and potassium rushes out, bringing the voltage rapidly back down in a process called repolarization, which resets the cell.
Once initiated, this electrical spike travels down the axon. And your body actually has a brilliant trick for speed here. In vertebrates, axons are coated in an insulating layer of fats and proteins called the myelin sheath. But this sheath isn't continuous. It has these tiny gaps called the nodes of Ranvier. So instead of traveling slowly along every single millimeter of the axon, the action potential literally jumps from node to node. This is called saltatory conduction. It allows the signal to travel at up to 268 miles per hour in your body. This massively increases speed while saving the cell a ton of energy. Finally, we reach step 5, crossing the synaptic cleft, bridging the physical gap. So the electrical signal has sprinted all the way down the axon, but eventually it hits a physical wall. The neuron doesn't actually touch the next cell. The electrical signal cannot jump across this tiny 20 to 30 nanometer physical gap, what we call the synaptic cleft. Because of this, the communication must instantly transform from an electrical sprint into a chemical flee. The end of the axon contains these tiny storage packets called vesicles that are just absolutely loaded with chemical payloads known as neurotransmitters. Here is the rapid-fire sequential chemistry that bridges the gap. First, the action potential arrives at the terminal bulb. Second, the voltage change opens specific gates, allowing calcium to rush into the cell. Third, this sudden entry of calcium triggers a process called exocytosis. The vesicles fuse with the outer membrane and just burst open, spilling their neurotransmitters directly into the synaptic cleft. Fourth, these newly released neurotransmitter molecules float across that microscopic gap and attach to highly specific receptors on the receiving cell's membrane. But they don't just attach anywhere. They click into these receptors exactly like a physical key fitting perfectly into a specific lock. Once they click in, they open ion channels on the new cell, creating either a new EPSP or IPSP. And just like that, this exact moment triggers the next cell to start the entire relay race all over again. But of course, you can't just leave those keys in the locks forever, right? The signal has to be swiftly terminated so the whole system can reset. The body handles this in a few really clever ways. The presynaptic neuron might vacuum the chemicals back up to recycle them, which is a process called reuptake, or specialized enzymes in the gap might just break the chemicals down completely. The releasing cell also has autoreceptors that act kind of like a thermostat, detecting when enough neurotransmitter has been released so it can shut off the supply. The receiving cell can even send retrograde transmitters, basically reverse chemical messages telling the first cell to stop.
So let's bring this all the way back out to the macro scale. The very next time you get a stressful text message and feel your stomach completely drop, remember what's actually happening inside of you. That feeling isn't magic. It's millions of microscopic bowstring snapping, sodium rushing in, electrical signals jumping across nodes of myelin at hundreds of miles an hour, and chemical keys unlocking receptors. Every single human thought, memory, and emotion is ultimately built upon this microscopic, highly choreographed dance of ions.
Which leaves us with something pretty profound to chew on. If everything we experience, every moment of joy, every complex problem we solve, every sudden feeling of panic, boils down to sodium and potassium crossing a tiny cell membrane, what exactly does that mean for the nature of human consciousness itself? Are we really just the sum total of our action potentials? Thank you so much for joining me on this explainer. Keep wondering, keep learning, and I'll catch you on the next one.